Now students, look at this. This is PBR 322. Okay, so you have the tetracycline gene over here, tetracycline resistance gene, and then you have the AMP, that is ampicillin resistance gene over here. Now there is a site for a restriction enzyme that is called BAMH1. So what we are going to do is, suppose I am using red color for this, your desired gene you need to introduce into this PBR 322. So where am I going to exactly introduce this desired gene? I will introduce at this site of tetracycline resistance gene, where exactly the site for cutting off the DNA BAMH1 resides. So actually I am removing the gene that is responsible for resistance to tetracycline. And now I am introducing my desired gene into that space. Now what would have happened to this PBR 322? It would have lost its resistance to tetracycline. That means if I add tetracycline to this plasmid, and if this plasmid is inside a bacteria, that bacteria will die. Now if I am going to introduce this particular plasmid into an E. coli and I am going to add ampicillin, that bacteria will live because ampicillin resistance gene is intact. What I have removed here? Only tetracycline resistance gene at BAMH1 site, and I have introduced my resistance gene over here. Now if you ask me why we are doing this, because I need to isolate and eliminate the non-transformants. Non-transformants are the ones which have not taken your desired gene. So there is no desired gene in non-transformants, or you can also call as non-recombinants. This is not needed for my biotechnology procedures. Now what I have to do, I have to take only the transformants that contains my desired gene. Those that have the desired gene intact, only that I will be having, and the ones that have not taken up my desired gene will still be having tetracycline resistance intact in them. So even if I add tetracycline to it, those bacteria will be able to survive in tetracycline medium because your desired gene is not taken up by the plasmid here. But here transformants would have taken up the desired gene, thereby they would have lost the tetracycline resistance, and if you add tetracycline antibiotic into that, those bacteria will die. So this is the concept. I'll repeat once again. In PBR 322, if you are going to add your desired gene in the tetracycline resistance gene site, those transformants would have lost tetracycline resistance. So they will die in tetracycline medium because in that site they have your desired gene. But at the same time, ampicillin gene, you have not inserted any of your desired gene. So that is intact over here. Now students, listen, we are going to do the next step, that is we are going to take handful of E. coli. We do not know how many of the E. coli would have taken that PBR 322. If they had taken the PBR 322, that means they are transformants. Those which have not taken up PBR 322 will be what? They will be non-transformants. Why? Previously we have seen the one that contains your desired gene are having the tetracycline resistance lost in them. So the ones which have not taken up your desired gene will be still having what? Tetracycline gene intact. That is, the resistance to tetracycline is still intact in them. So now see, I am going to take this handful of bacteria and I am going to put it in a plate, and I call this plate as master plate. Okay, and now This plate is nothing but a Petri plate where I have added agar. And in this agar medium, I am going to plate this bacteria. So E. coli will start growing like this. So this is how E. coli grows on a plate of agar. Now listen students carefully. This is called as master plate. This is where in ampicillin medium both transformants and non-transformants will be able to grow. Because in transformants and non-transformants, you can see that the ampicillin resistance gene is intact. And this master plate contains agar that is mixed with ampicillin antibiotic. So this medium is containing ampicillin in them. So even if you are going to give ampicillin in this medium, since transformants and non-transformants have both the ampicillin resistance gene intact in them, they will be able to grow. They will not be killed by the ampicillin antibiotic present in this medium. Now if you see the next plate, what are we going to do? This is called as replica plate. Now let me come back to this. This entire procedure is called as replica plating. What am I going to do here? In one plate I've added ampicillin along with agar. Another plate I'm adding tetracycline along with agar. So I'm mixing a powder of tetracycline antibiotic with agar and I'm going to put it in this plate now. So one plate contains ampicillin, another plate contains tetracycline. Now how do I transfer these colonies onto this colony is that I'm going to do a replica. So guys, I'll show you this. This is nothing but it is a stamp. Okay, so that is how the equipment will be in a lab. In a typical biological lab, biotechnological lab, they will have this. So you just have to keep it on top of this ampicillin medium and just touch the colonies on top of it. Just touch them. So imagine this is going to be a round stamp like structure. And now what you're going to do, without even changing your hand or the position or the orientation or direction, you're going to just take it and just keep it on the tetracycline medium. Just touch and then take off your stamp from here. So what you have done now, you have transferred a bit of colonies on top of this from ampicillin medium to tetracycline medium. I want you to listen to the last step carefully. Now that you have transferred the colonies from ampicillin medium to tetracycline medium, what do you think will grow in the tetracycline medium? Those bacteria which have their tetracycline gene intact. That means those bacteria which are non-transformants will grow in this medium. Why? Because here we have added tetracycline as the antibiotic here. So if you are going to grow a normal bacteria, they will die. If you are having a non-transformant with the tetracycline gene intact, that will be able to grow here. But if your transformant that contains the desired gene is put in this medium, do you think it will grow? It will not grow. Why? Because you have removed the tetracycline resistance gene and you have inserted your desired gene. So definitely it will not grow here, they will die. So in this medium, what you can see is those bacteria which are non-transformants, only that will grow in this medium. So this is how you will get. So all this that you see here, the green color ones, they are the non-transformants. Maybe I could give a different color for it. So these are the non-transformants. Now what we are going to do now, we are going back to the master plate. Now this is a non-transformant, so we can take this as non-transformant, non-transformant. This is non-transformant. So you just have to compare these two plates. That is why they say it is a cumbersome procedure, because we are comparing two plates over here. So now I have marked everything here. Whatever has grown here is a non-transformant. Now I am going back to the master plate from the replica plate and I am finding only the non-transformants. So these are the ones I do not need for my biotechnological procedure. So remaining ones are these. These are the transformants. I'm just marking all this because they have grown in this medium. But they did not grow where? They did not grow in this medium. So these are the spaces I am identifying. Here you would find only dead bacteria. These are all dead. These are all dead. They cannot grow. Because you have inserted your desired gene, so tetracycline resistance gene is lost, so they will die in tetracycline medium. Now what you have to do, you have to go back to your master plate and then you can retrieve all these and you can be very, very sure that these are the ones which are the transformants. These transformants are the ones which contain my desired gene, and only these will be taken and it will be propagated. So the ultimate aim of biotechnology procedure is I am going to propagate only my desired gene and I am going to eliminate all the undesired ones. So elimination is happening over here. Here also elimination is happening because I don't want all this. These are all the non-transformants. And I am going to select only the transformants for my procedure. So this method is called as replica plating. Indeed, this method is very, it is a cumbersome procedure because we need to compare two plates and we have to retrieve the ones which have recombined. Hope you are able to understand, children. Thank you.